Thank you, Amy, for that introduction. I'm very pleased to be with you today. We're talking about congenital heart disease, and congenital heart disease is really complicated. I think many pediatricians realize this. The reason I say it's complicated is it's complicated in two dimensions. First is the actual anatomy of the heart, and what we have to do in terms of surgical repair and long-term cardiac care for those patients can be complicated. And that's because long-term we can have issues of heart failure even after the corrective surgery, problems like arrhythmia, pulmonary hypertension. But for some individuals, surgery is not one and done. It's a lifelong condition that we have to continue to manage and maintain. In addition to the heart, although the cardiologists certainly are focused in that, as pediatricians, we also worry, of course, about the whole child. And the whole child can come with other issues with growth, sometimes trying to figure out whether or not those growth issues are related to heart failure or are independent. In some cases, other birth defects or congenital anomalies, which themselves can have other medical implications. And as well as being the heart that's involved, the brain is an important part about this. So everything we see above the shoulders has important day-to-day implications for the patient and their family. In some cases, these can be medical issues like seizures or epilepsy. In other cases, as the child is older, there can be things that we see, especially in school, issues with attention, specific learning disabilities. And as individuals get even older, things that may be coming up in terms of behavioral or neuropsychiatric conditions, things like obsessive compulsive disease or disorder, depression, and issues with executive functioning. And I'll talk about it a little bit, but there's also an increased risk of autism spectrum disorder for individuals with congenital heart disease. So one of the issues that comes up quite frequently is with newborn screening, for instance. We're identifying within the first few days of life children that have cyanotic congenital heart disease. And one question parents are very commonly asking is, so what should I expect from my child? Long term, what can we expect in terms of going to college, living independently, life expectancy, potential disabilities? And it's difficult because by even the best echocardiograms, even with the best CT surgeons and cardiologists talking about the cardiac prognosis with repair, we oftentimes at this point have difficulty saying what the long-term prognostic implications are for all of these other things that are so important to the day-to-day lives. So as we think about this, we've been trying to think about genomics, genetics, genomics, how we can use that information to get more precise information about prognosis and ultimately to support that patient and their families even better. As we think about this, just to appreciate that with newborn screening, we're talking about this phase of life. We're also even appreciating before babies are born in some cases, based on fetal echocardiograms and ultrasounds, in certain cases that we will have a child that when born will have structural heart disease. And so this is also within this continuum of life that I want to acknowledge that we also are doing things on the prenatal side as well as in the neonatal or postnatal side. And so all of these pieces come together, but to support the same hope in terms of the best chance at a healthy life for these children. So what is the role of genomics then in terms of this part of the picture? Part of it is to be able to get a more specific diagnosis, as I said, and to do it in some cases as quickly as possible to inform the prognosis, to be able to help parents rather prepare for future treatments, other comorbidities, other expectations. There are some people who don't like surprises, who like to be able to plan and prevent. And when we know what to monitor for in terms of health issues, we hope that we can prevent some of these issues down the road. And this would include, by the way, early intervention, again, giving a baby the best chance at a healthy life by starting, whether it be physical therapy, occupational therapy, speech therapy, but things where we have opportunities with the developing brain to be able to get in early and hopefully be able to maximize the outcome. To a certain extent, although this is not directly for the child with the congenital heart disease, families are often concerned about future risk to other family members if they're still in their reproductive years, thinking about other children, thinking about the risk of recurrence, if there are particular opportunities to take certain measures, be they prenatal diagnosis with fetal echocardiograms, specific genetic diagnoses, or even pre-implantation genetic diagnoses. And then I'll also say, and I find this more with mothers, but mothers and fathers asking the question of, just cosmically, why did this happen? How did this happen? What was the underlying cause? Was there something, moms often ask, was there something I did during the pregnancy that could have caused this? And addressing this issue of why our family, and in many cases, as I'll explain in a second, being able to absolve parents of guilt. And although that might seem small, I think that's actually in some ways a very big load off parents' shoulders. So let me try and put this within the context of an emerging picture of what we're understanding with congenital heart disease. You'll hear me say it again and again, but it's complicated. I think we've made incredible progress over the last decade, but we are very far from understanding this either in terms of all etiologies or even just the genetic etiologies. I'm going to try and paint a picture, and I will say this picture is true for many complex conditions, be they congenital heart disease, other birth defects, even autism and other neurodevelopmental issues. What I'm going to focus a lot on today is this big blue box. And what I mean by this big blue box are single genetic events that are what we call highly penetrant. That is, that if one has this genetic event, there is a very high risk of developing whatever that condition is. When I say very high risk, I'm on the order of, for instance, 50-fold, 100-fold increased risk. So this is not a teeny tiny little genetic thing. This is a powerful genetic factor that's influencing the risk. That's at one extreme. This is what we, at this point, understand the best with congenital heart disease and related disorders, but it's not the only part of the answer. In addition, at the other extreme, there are things that you may have started hearing about, which we call polygenic risk scores. In contrast, this is not one single genetic event, but oftentimes hundreds, even thousands of genetic contributors, each one of which contributes a very small amount of the risk. And when I say small, that could be a relative risk of 1.1 or 1.05, but a very small amount of risk. This is the type of contributor we see to things like height, where it's not just one genetic factor for being tall, but many, many contributing genetic factors. And this is more hypothetical at this point, but we do think this could play an important role for structural birth defects like congenital heart disease. In the middle of this are factors that are moderately penetrant. These may confer risk of something on the order of, for instance, 5-fold increased risk, certainly much higher, but not sort of these one strong factors. In these cases, we think it may take not just one genetic contributor, but maybe 2, 3, 5, but some countable number. In reality, at the end of the day, when it comes to traits that we see like intelligence or behavior, complicated traits, it really is a composite of all of these factors coming together in that individual person. And that's why sometimes you'll hear geneticists say, even for a single highly penetrant genetic factor, we can't perfectly predict the prognosis. We can give a range of possibilities, but not perfectly predict the prognosis because it's in fact determined by some of these other both oligogenic, moderate penetrant genes, as well as these polygenic factors as well. All of those come together to give the most refined prognosis. Okay, so let me focus on one very important class of genetic changes, and those are de novo genetic changes. So many times you'll hear from parents, well, I don't see how this could be a genetic factor in our family. We have no history of congenital heart disease. Well, we now realize that at least some fraction of congenital heart disease is due to de novo genetic changes that are present in the child. So they are in the genes in the child, but they're not present in the mother or the father. That is, it started brand new. And when I say not present in the mother or the father, what I mean by that is when we take a blood sample, a saliva sample, we don't see it in those cells in the mother or the father. Where did this happen then? Where did it come from? Well, we don't know based on the genetic testing we do oftentimes exactly when that happened, but we hypothesize that it was present either in a small number of the gonads of the father or the mother, so in, for instance, one sperm cell or a small number of sperm cell or egg cells, again, not in the blood of the parent, but in just the gonads, or shortly after fertilization, a mutation that arose in that developing embryo, but very likely very, very early on in development. That's very important to know because, as I say, then we don't have to worry about the blame game. This was not something either parent could have done to prevent this problem. It was not something that someone was responsible for. It doesn't have to do with the mother having a glass of wine before she realized she was pregnant, missing a day of her prenatal vitamins. There's no fault that comes with this. It also means that in terms of recurrence risk for these parents, the likelihood that it will happen again for a future child, if it was de novo in this child, is usually less than 1%. And we've determined that empirically because about 1% of those parents will have some other gametes, either sperm or eggs, that have that same genetic change. But to me, that's a low recurrence risk for the parents. Now, on the other hand, for this child, when they grow up, because this is something that is now theoretically and very likely in all of their egg or sperm cells, their risk of recurrence is very likely to be something like 50%. So important difference in terms of recurrence risk depending on who you are in the family. So as the data that I'm going to show you, this is based on some large studies that have been done by a group called the Pediatric Cardiac Genomics Consortium, a group that's funded by NHLBI that I've been fortunate to be a part of for the last 10 years. And it's based on very robust evidence. Certainly, we still have more to learn, and we're still working very hard within what I'll call for short the PCGC, but it is robust, and it's based now on what we call genomic analyses that include exome or genome sequencing, as well as genome-wide genotyping on almost 5,000 individuals with congenital heart disease, as well as their parents. So a very robust data set. Within this, we have been able to, as I said, appreciate by studying the parents and the child that de novo genetic events are important. So what did we do within the PCGC? We did things like exome or genome sequencing, very comprehensive ways of looking at the genomes. We did this for the mother and the father and for the child with congenital heart disease. We could very quickly filter out through all of those variants, which number in the hundreds of thousands. So realize a lot of genetic variation, be able to go through that and see which were those genetic changes that were new in the child and not present in the blood sample from either of the parents. On average, each child has about 100 of those genetic changes, somewhere between 100 and 200 throughout the genome, and approximately 1 to 2 that are in the coding sequence within their genome. So that now brings the search space down tremendously, and it was why we set the experiment up this way. I will tell you, I've done this experiment with my own children, and all of us who have had children have de novo genetic changes in our children. That's not what's unique, and that's not what's special with individuals with congenital heart disease. What is special is where those de novo genetic changes fall. They're not randomly distributed, but they're enriched for particular types of variants that are more likely to be deleterious or cause problems with the way those genes function. And they're more likely to be in sets of genes that are very much responsible for how the heart develops. And that's why they lead to congenital heart disease. One of the things we learned from these first studies that were published in Science and Nature was that things are complicated. We predicted that there would be at least 500 different genetic changes, each of large effect size contributing to congenital heart disease. So you can already understand the heterogeneity within this one umbrella term that we call congenital heart disease. As we started looking at those de novo genetic events, we realized that they started to help us understand prognosis because they were associated with a particular group of individuals with congenital heart disease. So let me walk you through this. Shown here are the percentage of individuals by phenotypic class or by particular traits. So as I'm showing you, control individuals who have no congenital heart disease, no other medical issues, they do have a certain fraction of these de novo genetic variants, but they're relatively low. If we look at all individuals with congenital heart disease, they're enriched approximately 8-fold over that. But now, if I start separating out individuals with congenital heart disease to those who have isolated congenital heart disease, that is, no other birth defects or no other congenital anomalies, that difference or that you can see an enrichment for those individuals who have congenital heart disease plus another anomaly. So that's shown here in blue as well as shown here in red. If I now compare that to individuals with congenital heart disease and who also have a neurodevelopmental disorder, so it could be something like seizures or autism or intellectual disabilities, I do see, again, compared to isolated congenital heart disease and enrichment, or more of these individuals have these de novo deleterious genetic events. Up to 20% of individuals who have congenital heart disease, plus another congenital anomaly, plus a neurodevelopmental condition, up to, on average, 20% of those individuals have these de novo genetic variants that we think are contributing significantly to their congenital heart disease. Since we've done those initial studies that are now almost 7 or 8 years old, we've continued to look at that and to fill out this space, not only for de novo genetic events, but also for inherited genetic events. And I've broken these down into de novo copy number variants. So those are not single genes, but contiguous gene disorders. Those with de novo genetic events due to likely gene disrupting single gene disorders, as well as those with deleterious missense genetic variants shown in each of these classes, as well as now just starting to understand rare transmitted or inherited genetic events that are likely gene disruptive. What you can see is that for individuals with isolated congenital heart disease, we see approximately that we can account for about 21% of those individuals due to these strong genetic events, either copy number variants or single genes. On the other hand, approximately twice as many individuals with complex congenital heart disease, complex because they have either a neurodevelopmental disorder and or another birth defect, approximately 42% of those individuals have one of these strong genetic events that are contributing to the congenital heart disease. So in other words, we're now starting to understand a significant fraction. And in the newborn period, we oftentimes don't know for a baby if they are in this bucket of isolated or complex. So at the very beginning of life, before we started to get to know that baby, on average, we'll see that about a quarter of those individuals will have one of these genetic events, and that helps us to predict prognosis. Again, potentially the need to look for other medical issues or to anticipate neurodevelopmental issues. Now, to understand some of these inherited events, these are important, especially for parents to know, because these have a higher risk of recurrence, in some cases up to 25 or even 50% risk of recurrence. We've now started to appreciate autosomal recessive conditions that have their own spectrum of conditions associated. Many of these are what we call ciliopathies, and those are in particular associated with respiratory or lung conditions. So we need to be especially careful with that group of children in terms of pulmonary conditions, breathing problems, lung infections. And then we've also started to identify other genetic forms that I won't have time to go into today, but they have their own specific prognosis. We've even identified what we call founder mutations, or specific mutations that may be specific to individual communities. And just one example of this is a gene called GDF1, for which we see a very specific type of congenital heart disease, transposition of the great arteries and double outlet right ventricle, and one mutation that's specific within the Jewish community. And so this helps us to be able to anticipate and think about even targeted testing in certain communities. I won't go into it too much today, but we are starting to now understand the space of non-coding genetic variants, and in particular, de novo non-coding variants. This is still very much an area of exploration. I don't think we're ready for clinical translation of this information yet, so I'm not going to go into a lot of detail, except to say that these types of genetic variants, the risk conferred, or that odds ratio on the left, is far less than, for instance, 10 or 100 or 50-fold. It's more on the order of 1.2 to 1.5. It's a modest or oligogenic risk as we think about this, but maybe important in the larger scheme of things. Overall, as we're seeing this, we do know, again, the take-home picture that de novo non-coding variants are contributing to congenital heart disease, but we do anticipate that rather than some of the studies that are now powered on approximately 500 or even 1,000 samples are woefully short of the sample size that we'll need to understand those. So this is an area to watch for the future. We do realize, as I said, that some of the genetic changes may be happening post-zygotically. That is, that the genetic change may not be in the egg or the sperm, but may be after the point of fertilization, and as a result of that may lead to mosaicism. And again, this is a relatively newer area for us, but as we've done this, we realize that that prognosis may be a little bit different because not every cell in the body has that genetic change. And in particular, it depends what cells in the body are affected and what percentage of those cells. And it becomes difficult to infer prognosis as much as we'd like to. And that does produce some challenges with confidence in what we know, but we are increasingly appreciating that that is a source of some of the genetic changes we see. As we've done some of these studies of mosaicism, again, I think we're just starting to understand what this is, but we have the sense that it could be as many as 1 in 8 patients with congenital heart disease has a mosaic change. Some of these we may not even be detecting with our usual methods of clinical diagnostic testing because the levels that we see in the blood or in saliva are so low that we may not be doing the test in the right way to be able to detect that. Over time, I anticipate we'll develop tailored tests to be able to see those. So let me switch gears to talk about prognosis. Once we have the information about the genes, how does this help us in terms of thinking about prognosis? I'm going to start by taking overall, and again, thinking about newborn screening, thinking about those babies when they're in the neonatal intensive care unit. The bottom line is that when we think about all babies with congenital heart disease in the NICU, those that had positive genetic test results or those that had negative genetic test results, perhaps not surprisingly, what we see is that from a percentage point of view, those who had positive genetic tests had a higher frequency of complications in the neonatal period, had a higher median length of stay, and had a higher percentage of neurodevelopmental issues long term. And these are statistically significant. So we know that those babies are more complicated and helps in terms of planning expectations. This is now looking at a Kaplan-Meier survival curve, so looking at time on the x-axis and survival probability on the y-axis, and looking specifically at copy number variance after surgery. And so either if we look at death or we consider cardiac death or need for cardiac transplant similarly, we know that individuals shown in red in this dotted line have a more serious long-term prognosis, that is that they are more likely to die or to need a heart transplant than individuals similarly paired with the same types of congenital heart disease but without copy number variants. So again, part of what we hope to do is identify those who have the copy number variants, hopefully intervene by setting expectations in terms of associated medical conditions and getting in proactively and preemptively to treat those conditions, to ameliorate those complications, to prevent those problems that ultimately led to this worse life expectancy and worse survival. So knowing early and acting should be helpful. Again, one of our tenets of newborn screening. When we also think about this now for not just single genes, or rather not for copy number variants, but looking at single genes, this is similar looking at neurodevelopmental outcome, and in red are the individuals that had likely pathogenic genetic variants. You can just look at these across these different categories, which I won't get into too much today. And in blue for comparison, those individuals with congenital heart disease without those genetic variants, what you can see is that there is a higher prevalence of neurodevelopmental issues across those groups in red. And then in a similar sort of way, if we look at now just the heart, so not looking at the brain, but looking at the heart, similarly, if we look at systemic ventricular function, so how well the heart squeezes, if we look at this in terms of over time, longitudinally worsening or dynamically ventricular functioning getting worse, or arrhythmias, what we can see is as a class, those individuals who have genetic events have a higher frequency of these cardiac complications. And these are specifically, I should point out, for individuals with single ventricles. So one very specific type of congenital heart disease, we're now able to give prognostic information that may help us, for instance, in terms of monitoring for arrhythmias, monitoring more carefully for decreases in heart function, so that we can think about who might need to be listed for transplant, who might need to be for instance, defibrillator, who might need medications for heart failure, but being able to anticipate those individuals. Even when it comes to the complication of pulmonary hypertension, we are now starting to define the specific genes within all of those large number of genes accounting for congenital heart disease, which are the genes specifically that portend a higher risk of pulmonary hypertension. And so again, when we get back those genetic test results, if you have one of these genes, we may need to follow more carefully, even by right heart catheterization, or at least looking very carefully at the right-sided heart, part of the heart, what the pressures are like for the pulmonary circulation. So as we've been doing this, I think one of the luxuries I've had is, as an investigator, being able to search and study multiple different conditions. So I've been telling you about congenital heart disease, and although it's not part of newborn screening, at least not at this point, I also study conditions like congenital diaphragmatic hernia, or CHD, on the left, as well as neurodevelopmental disorders on the right, shown as intellectual disability, autism spectrum disorder, neurodevelopmental disorders. And what I'm trying to show you here is that for each of these genes on the left, I've broken them down into major categories, be they transcription factors, chromatin modifiers, post-transcriptional regulators, protein kinases. What I'm trying to show you here is that many of these same genes actually confer increased risk across birth defects as well as across the brain. Now, it's not every single one. So you'll see that if you look across a row, not all genes. GATA6 doesn't necessarily have intellectual disabilities associated with it. We see this more based on how the body develops in terms of the diaphragm or the heart. On the other hand, the majority of these genes that I'm showing you within this table, actually you'll see dots for the row going across, meaning that individuals, at least certain individuals with these genes, have been identified that have intellectual neurodevelopmental issues, plus or minus other associated birth defects. So now going back to what I was telling you before, what's interesting, and one of the challenges that we have, is that once we identify individuals with changes in one of these genes, how can we now even fine-tune within that gene which individuals will have which manifestations? We haven't figured that out yet. I think that's when we get into the space of not just that big blue box I was showing you before, that might be this major gene that I'm showing you here, but how do we look at other interacting factors, some of those tiny little gray boxes or some of those medium-sized boxes that now interact with these major genetic drivers to determine for any one individual what their manifestations will be with that condition. And that's part of what we're studying now. Part of the way we're studying that is to aggregate individuals from around the world that have the same genetic change. So this is one of many communities I've had the pleasure of working with. This is one particular group, PAX1, and this is a photo from our first in-person family meeting a few years ago. And I have permission to share these photos with you. These individuals all have the same PAX1 gene, and believe it or not, they have the same exact change within the PAX1 gene. So that big blue box for every single one of these children I'm showing you here is the same. On the other hand, we appreciate within the PAX1 community that there's still variability. This particular gene is associated with congenital heart disease, as an example, but not every one of the children on this picture has congenital heart disease. So what are those other factors that are also influencing ultimately who has congenital heart disease? And that's the next wave of what we're working on now, again, with very large international consortia. One of the challenges, though, is for each one of these rare genetic conditions, we estimate that the prevalence is probably about 1 in 100,000 births. So they're individually rare, collectively common, because congenital heart disease is common, but individually rare. So to wrap things up, I hope you have the impression that genes are important when we come to thinking about congenital heart disease. This is very much a work in progress. We don't have this all figured out, but we are getting to the point where we know enough that we are starting to clinically implement these tools. So what I've shown you about in terms of using chromosome microarray, EXOME, or genome sequencing, These are clinically available tools. This is not just being done within research studies like PCGC. These are clinically available. We hope that knowing this information will help us in terms of anticipating and preventing problems. We still do not yet have a way of getting at the underlying genetic change and being able to do gene surgery and fix the genes or correct the genes or put in the missing genes. Maybe someday we'll get there to that point, but right now it's really focused on being able to help empower families, help set expectations about what to anticipate, and with their providers working closely with them to anticipate, prevent, and ensure the best chance at a healthy life for these children. I would be foolish to think that was my work, so I want to be very clear. This is a team effort. We do a lot of team sports and team science, and I have the pleasure of working with many talented individuals. This is my crew at Columbia, but just to realize this is powered really by families, and to acknowledge the incredible contribution that families make to us shoulder to shoulder within all of these phases, along with our multidisciplinary groups of bioinformaticians, cardiologists, neonatologists, really being able to work together. So I will stop there, but thank you for your attention, and I'll be glad to take questions.